Hello everyone, this is Wuwei from Dr. Yang Shen's lab, Electrical Engineering Department, Texas A&M University. I'm very happy to present our work non-coding variant effect prediction using genome sequence and chromatin structure here today. The introduction of the background. The non-coding DNA are the genomes that do not encode proteins. Non-coding variants are related to lots of diseases. Current methods measure the non-coding mutation effects by computationally predicting the epigenetic profile changes. Those models rely on the sequence only. We like to incorporate both 1D genome sequence and the 3D chromatin structure. We can see the folded DNA sequence, like the topological assertion domain. We hope the far in 1D but closer in 3D genome information can help the epigenetic profile prediction. First, we validate the potential helpfulness of the 3D chromatin structure. We used one-sided case test to show the genome regions with dissimilar genome sequence but similar epigenetic profile are closer in 3D. Each subplot is using one cell line's chromatin structure. Each row is for one high-assay experiment. From left to right, we have increasingly dissimilar sequence and similar profile. The color is showing the significance of the case test. We can see for all three cell lines and most high-assay experiments, we have the significant case test result, which means low genome sequence but high profile similar regions are closer in 3D. So we hope the 3D structure can correct the inconsistency among genome sequence model input and epigenetic profiles model output. To incorporate 1D genome sequence and the 3D chromatin structure, we use the CN or RN to embed genome sequence and the fully connected layer or graph convolution layer to embed chromatin structure. Then the embedded information are concatenated for the final epigenetic profile prediction. We do not have a representative chromatin structure, so each time we used one cell line's chromatin structure to predict epigenetic events for multiple cell lines. Here we are showing the average AUPRC of the epigenetic events prediction. For all three cell lines chromatin structure, we always have better performance compared to the sequence-only model, and if we're using the RN with the GCN embedded topological plus sequence information, we can always have the best performances. And it's always better than the sequence-only, the left column. Next, we would like to see our model's performances on the real application. First, we'll focus on the cell line specific non-coding variance effect prediction. The variance and the chromatin structure are from the same cell line. We're predicting for a variant gene expression is enhanced or suppressed. Our model, the red one, is doing better than the sequence-only model, the blue one, especially for the variants with significant effect. The expression level fold changes greater than 2 on the right of the dashed line. Then we'll focus on the general EQTLs. We're identifying the grasp EQTLs against the negative SNPs. As shown in the figure, our model is doing well, especially when the EQTLs are far from the negative SNPs in 1D, which relies on our 3D structures information helpful. Next, we try to use our model to identify motifs. All three of our models missed 7 motifs identified by the sequence-only SOTA model. However, all three of our models find 2 motifs that the SOTA missed, and we find that for those 2 motifs related to POU3F1 gene and TFDP1 genes, both of them are closely connected to its collaborator in 3D. So we believe it's because we use the 3D chromatin structure information that helps the model to find those motifs. At the end, for the conclusion, we proposed an architecture to incorporate 1D genome sequence and 3D chromatin structure for epigenetic profile prediction. And we have shown the improvement of prediction accuracy come from the correction of inconsistency among genome sequence and epigenetic profile. At the same time, we have shown the proposed model is doing well in real applications and non-coding mutation effect predictions. Thanks for watching.